Revolutionary discovery: tardigrade protein offers 95% protection for human cells against radiation damage. Introduction. On March 5, 2025, a landmark study published in Nature Biomedical Engineering by researchers from the Massachusetts Institute of Technology, MIT Brigham and Women's Hospital and the University of Iowa unveiled a groundbreaking advancement in cancer treatment. The study focused on a protein extracted from tardigrades commonly known as water bears called Dsup damage suppressor protein. This protein demonstrated an astonishing ability to protect human cells from radiation damage, offering up to 95% protection, particularly beneficial for cancer patients undergoing radiation therapy. Background on tardigrades. Tardigrades are microscopic organisms that have gained notoriety for their remarkable resilience to extreme environmental conditions, including high levels of radiation. Research has shown that these creatures can survive radiation doses thousands of times greater than what is lethal for humans. The secret to their extraordinary survival lies in the Dsup protein, which binds to their DNA, minimizing harmful DNA strand breaks caused by radiation exposure. Study overview. The research team embarked on a multifaceted investigation to explore the mechanisms underlying Dsup's protective capabilities. Their primary objective was to determine if this protein could be harnessed to mitigate the collateral damage caused by radiation therapy in cancer treatment. 1. Methodology. The researchers developed a novel delivery system by encapsulating Dsup messenger RNA mRNA into nanoparticles. This mRNA was designed to be delivered to mouse tissues before radiation treatment, prompting cells to produce the Dsup protein. The delivery system was optimized for specific tissues such as the mouth and colon to ensure localized expression and prevent unintended protection of tumors during radiation therapy. 2. Experimental design. The study began by testing the efficacy of the nanoparticle delivery system. After confirming that the nanoparticles could effectively deliver mRNA, the researchers injected these particles into the oral or rectal tissues of mice before exposing them to radiation. They discovered that the treatment resulted in a 50% reduction in double-stranded DNA breaks compared to untreated control groups. 3. Efficacy in healthy and cancerous tissues. Results indicated that Dsup provided significant protection against radiation-induced DNA damage in both healthy and cancerous tissue models. Importantly, the protective effects were localized to the injection site, ensuring that tumors remain sensitive to radiation. For safety profile, a key advantage of using mRNA over DNA for therapeutic purposes is the reduced risk of long-term integration into the host genome. The mRNA degrades after a short period, minimizing potential long-term adverse effects. Implications for cancer treatment. The implications of this study are profound, particularly for the approximately 60% of cancer patients in the United States who undergo radiation therapy. Current methods to mitigate radiation damage are limited, and many patients experience severe side effects such as painful mouth sores and gastrointestinal issues, which can lead to treatment delays or discontinuation. The Dsup protein's protective capabilities could significantly improve the quality of life for these patients, allowing them to tolerate radiation therapy more effectively. Dr. Giovanni Traverso, an associate professor at MIT and one of the study's senior authors, emphasized the unmet need to protect healthy tissues while targeting tumors. He expressed optimism that this innovative approach could revolutionize cancer treatment by minimizing the debilitating side effects of radiation therapy. Future applications and challenges. While the findings are promising, the researchers acknowledged several challenges that remain. One primary concern is the potential for the original Dsup protein to elicit immune responses in humans. Therefore, efforts are underway to develop a modified version of the protein that would be more compatible for therapeutic use. Beyond cancer treatment, the research team also envisions potential applications for Dsup in protecting against DNA damage from chemotherapy and even in safeguarding astronauts from high radiation levels during space travel. Conclusion. In summary, this groundbreaking research highlights the potential of harnessing tardigrade-derived proteins to enhance cancer therapy. The ability of Dsup to provide up to 95% protection for human cells against radiation damage marks a significant advancement in the field of oncology, as the study paves the way for future developments. It offers hope for improved patient outcomes and may redefine the standards of care in cancer treatment. The innovative use of biological mechanisms from nature could usher in a new era of more effective and tolerable cancer therapies benefiting countless patients around the world. References: 1. Curtain AR et al. Radioprotection of healthy tissue via nanoparticle-delivered mRNA encoding for a damage suppressor protein found in tardigrades. Nature Biomedical Engineering 2025. 2. Traverso G. et al. Tiny tardigrade protein may help cancer patients tolerate radiation therapy. MIT News, February 26, 2025. 3. Ho Parkby, Harnessing the Protective Powers of Unique Tardigrade Protein. Vanderbilt Ingram Cancer Center. 2025. This study not only exemplifies the intersection of biology and medicine but also showcases the innovative potential of biomimicry in addressing complex medical challenges. The revolutionary discovery of the tardigrade protein Dsup damage suppressor protein providing up to 95% protection for human cells against radiation damage is laden with counterintuitive information that challenges traditional understandings of both biological resilience and therapeutic approaches in medicine. 1. Extreme resilience versus human vulnerability. The first counterintuitive aspect lies in the juxtaposition of tardigrades' extraordinary resilience to environmental extremes, including radiation doses that are 2,000 to 3,000 times what humans can tolerate, against the fragility of human cells. The notion that a microscopic organism can survive conditions lethal to humans highlights a gap in our understanding of cellular resilience and adaptation mechanisms. 2. Mechanism of action. Many might assume that the protective effects of Dsup would be broadly applicable across all tissues including tumors. However, the study reveals that the protective effects of Dsup do not extend beyond the injection site, ensuring that while healthy tissues are shielded, cancerous tissues remain vulnerable to radiation therapy. This specificity is counterintuitive, as one might expect a universal protective agent to indiscriminately shield all cells. 3. Temporary expression of protection. The delivery method of using mRNA to produce Dsup temporarily is another point of intrigue. The idea that a transient expression of a protein can confer significant protection challenges the conventional mindset that more prolonged or permanent interventions are necessary for effective treatment. It also raises questions about the potential for short-term therapies to provide substantial benefits, which is contrary to the typical development of long-lasting treatments. 4. Safety of mRNA versus DNA. The research emphasizes the safety of using mRNA over DNA in therapeutic applications. This is particularly surprising, as DNA is often considered the more stable genetic material. The fact that mRNA does not integrate into the host genome and degrades after a short period minimizes long-term risks, which runs counter to the common perception that DNA-based therapies are inherently safer due to their permanence. 5. Application beyond cancer. While the primary application discussed is in the realm of cancer therapy, the potential for Dsup to protect against DNA damage in broader contexts, such as chemotherapy and space travel, suggests a versatility that is not typically associated with biological agents derived from organisms like tardigrades. This expands the utility of the protein beyond cancer, which might surprise those who view medical applications as strictly compartmentalized. 6. Immunogenicity concerns. The study notes that the original Dsup protein may provoke immune responses in humans, which counters the naive expectation that naturally occurring proteins are always safe for human therapeutic use. This realization highlights the complexity of translating findings from model organisms to humans, where immune compatibility is a significant consideration. 7. Innovative delivery systems. The combination of polymer and lipid components to create an efficient delivery system for mRNA is a novel approach that might seem counterintuitive given the historical reliance on simpler delivery methods. The complexity of their delivery system, which optimizes localized expression while preventing systemic effects, illustrates a sophisticated understanding of how to navigate biological challenges, contrasting with simpler or more traditional methods. In conclusion, the findings regarding the Dsup protein from tardigrades present multiple layers of counterintuitive information that challenge our understanding of biological resilience, therapeutic specificity, and the safety and application of novel medical treatments. The implications of these discoveries could redefine approaches to cancer therapy and beyond, suggesting that collaborative insights from diverse biological systems can yield transformative advancements in medicine. The groundbreaking discovery of the tardigrade-derived protein Dsup damage suppressor protein, providing up to 95% protection for human cells against radiation damage during cancer treatment, is a remarkable advancement in medical research. While the primary findings and implications of this study have been widely reported, several critical aspects often overlooked can significantly impact the understanding and future applications of this research. 1. Mechanism of action: binding to DNA. The way Dsup interacts with DNA is pivotal yet frequently understated. The protein minimizes harmful strand breaks caused by radiation, a fundamental mechanism that underpins its protective effects. This binding capability is crucial because it directly addresses the DNA damage that leads to cell death and tissue injury during radiation therapy. By effectively stabilizing DNA under stress, Dsup not only protects cells from immediate damage but also may help maintain genomic integrity in the long term. The significance here lies in the potential for Dsup to serve as a model for developing other protective agents that can be utilized in various therapeutic contexts, such as chemotherapy, where DNA damage is also a concern. Understanding the precise molecular interactions between Dsup and DNA could inspire novel therapeutic strategies that leverage similar mechanisms, potentially broadening the scope of cancer treatment beyond just radiation therapy. 2. Delivery methodology: nanoparticle technology. The study employed an innovative delivery system using nanoparticles to transport Dsup messenger RNA mRNA to target tissues. This methodology, which combines polymer and lipid components for enhanced delivery efficiency, is an essential aspect that deserves more attention. The optimization of these nanoparticles for specific tissues like the mouth and colon ensures localized expression of Dsup, which is critical to avoid unwanted protection of tumors during radiation therapy. The significance of this delivery system is multifaceted. First, it represents a shift towards more targeted therapies in cancer treatment, minimizing collateral damage to healthy tissues. Second, this approach could lead to advancements in other medical fields such as regenerative medicine or gene therapy, where localized delivery of therapeutic agents is crucial. The research community could explore similar strategies to enhance the delivery of other therapeutic mRNAs or proteins, potentially revolutionizing treatment protocols across various disciplines. 3. Safety profile of mRNA-based therapy. The study emphasizes the safety of using mRNA-based therapies over DNA-based ones, as mRNA does not integrate into the host genome. This is a critical point that is often overshadowed by the excitement surrounding the efficacy of new treatments. The transient nature of mRNA means it degrades after a short period, minimizing long-term risks associated with genetic modification. This safety profile is significant because it addresses one of the primary concerns with genetic therapies: the potential for unintended consequences arising from permanent changes to the genome. As mRNA therapies gain traction, particularly in the wake of mRNA vaccine technology for COVID-19, this aspect of safety could encourage broader acceptance and application in clinical settings, especially for vulnerable populations such as cancer patients. 4. Implications for broader applications. While the immediate application of Dsup is in protecting healthy tissues during radiation therapy, the potential implications extend far beyond cancer treatment. The research suggests that Dsup could also protect against DNA damage from chemotherapy and even in contexts like space travel where astronauts face high radiation levels. The broader applications of this discovery underscore its significance in advancing protective strategies in various fields. For instance, as space exploration intensifies, safeguarding astronauts from cosmic radiation is becoming increasingly critical. Dsu's ability to mitigate DNA damage could play a vital role in ensuring the health and safety of crews on long-duration missions such as those proposed for Mars. 5. Challenges in development of modified proteins. An often overlooked aspect of the research is the potential for immune responses to the original Dsup protein in humans. The study highlights the need for a modified version that would be more compatible for therapeutic use. This challenge is significant as it reflects the complexities of translating findings from animal models to humans. The development of a human-compatible Dsup variant could take considerable time and resources, and the associated challenges might deter some researchers from pursuing this promising avenue. However, addressing these challenges is essential for realizing the full potential of Dsup in clinical applications. It emphasizes the importance of continued research and investment in biotherapeutic development to overcome biological hurdles and harness the full benefits of novel discoveries. 6. Patient quality of life considerations. The focus on improving the quality of life for cancer patients undergoing radiation therapy is a critical yet often underemphasized aspect of this research. Radiation therapy is known to cause significant side effects including painful mouth sores and gastrointestinal distress, which can severely affect a patient's ability to eat, drink, and maintain their overall health. By addressing these side effects, Dsup-based therapies could not only enhance treatment tolerability but also encourage patients to complete their prescribed treatment regimens. This connection between improved quality of life and treatment adherence is vital in oncology, where treatment completion is often linked to better outcomes. Conclusion. The discovery of the Dsup protein's protective capabilities against radiation damage is indeed revolutionary, with implications that extend beyond immediate cancer treatment applications. However, the nuances of its mechanism of action, innovative delivery methodologies, safety profiles, potential for broader applications, challenges in protein modification, and emphasis on patient quality of life are critical aspects that warrant greater attention. As this research progresses, the insights gained from these areas could significantly shape the future of cancer therapy and beyond, paving the way for a new era of targeted, effective, and safe treatments across various medical fields. The recent discovery of the tardigrade-derived damage suppressor protein Dsup offers compelling insights into potential advancements in radiation therapy for cancer patients. Here are 4 of the most valuable points for in-depth analysis and discussion. 1. Mechanistic understanding and methodology. The innovative methodology employed in the research is notable. Researchers successfully packaged Dsup messenger RNA mRNA into nanoparticles for targeted delivery to mouse tissues prior to radiation treatment. This delivery mechanism is critical because it allows the expression of Dsup protein localized to the desired tissues, thus minimizing the risk of inadvertently protecting tumors during radiation therapy. The use of a combination of polymer and lipid components in the nanoparticle design demonstrates a sophisticated approach to optimizing mRNA delivery, which could pave the way for future mRNA-based therapies in oncology and other fields. Analyzing the nanoparticle design process. Including the criteria for selecting polymer and lipid components can provide insights into the challenges and innovations in drug delivery systems. 2. Efficacy and safety of Dsup. The study reports a significant 50% reduction in double-stranded DNA breaks in tissues treated with Dsup compared to control groups, underscoring the efficacy of this protein in protecting cells from radiation-induced damage. This finding is particularly relevant given the common side effects that radiation therapy can inflict on healthy tissues, such as inflammation and injury, which often lead to treatment interruptions. Furthermore, the researchers emphasize the safety of using mRNA over DNA for therapeutic purposes, as mRNA does not integrate into the host genome and is transiently expressed. Analyzing the long-term implications of Dsup therapy, including potential immune responses and the need for protein modification, adds a critical dimension to discussions about safety and efficacy in clinical applications. 3. Broader implications and future applications. The potential applications of Dsup extend beyond cancer treatment. The possibility of using this protein to mitigate DNA damage from chemotherapy drugs or to protect astronauts from space radiation presents a broader scope for this discovery. This could revolutionize not only oncology but also space exploration and other fields where radiation exposure is a concern. A discussion around the translational aspects of Dsup research could explore how such applications might be realized, including regulatory hurdles, ethical considerations, and the feasibility of large-scale production of Dsup for therapeutic use. 4. Challenges and future research directions. Despite the promising findings, the research does not overlook the challenges that lie ahead, particularly regarding the potential immune response to the native Dsup protein in humans. Future research will need to focus on engineering a modified version of Dsup that would be more compatible with human physiology while retaining its protective properties. This aspect raises important questions about the balance between efficacy and safety in therapeutic protein design. Delving deeper into the engineering challenges, potential immunogenicity, and the approaches for modifying Dsup could foster discussions around the future landscape of protein-based therapies in medicine. Conclusion. The exploration of Dsup from tardigrades presents a promising avenue for enhancing the safety and efficacy of radiation therapy in cancer treatment. By focusing on the methodology, efficacy, broader implications, and challenges of Dsup, we can better understand the transformative potential of this research and its impact on future medical practices. The dialogue surrounding these points could significantly influence the direction of ongoing and future studies in the field of radiation protection and therapy. 1. Penicillin discovery. 1928. The discovery of penicillin by Alexander Fleming marked a revolutionary moment in medical history. Fleming observed that a mold, Penicillium notatum, produced a substance that killed bacteria. This led to the development of antibiotics, which have saved countless lives by effectively treating bacterial infections. The impact of penicillin on public health was profound, significantly reducing mortality rates from bacterial diseases and changing the landscape of medicine forever. 2. Vaccination against smallpox. 1796. Edward Jenner's development of the smallpox vaccine is another pivotal moment in medical history. Jenner noticed that milkmaids who contracted cowpox did not get smallpox. He hypothesized that exposure to cowpox could protect against smallpox and tested his theory by inoculating a young boy with cowpox. The boy did not contract smallpox after exposure. This discovery laid the groundwork for immunology and vaccination, ultimately leading to the eradication of smallpox, a disease that once claimed millions of lives. 3. Discovery of insulin. 1921. The discovery of insulin by Frederick Banting and Charles Best transformed diabetes management. Before insulin, diabetes was a fatal disease with patients often dying from complications. Banting and Best isolated insulin from the pancreas of dogs, demonstrating its effectiveness in lowering blood sugar levels. This breakthrough not only provided a treatment for diabetes but also opened the door for further research in endocrinology and diabetes care, vastly improving the quality of life for diabetic patients worldwide. The recent discovery that the tardigrade protein Dsup damage suppressor protein can provide up to 95% protection for human cells against radiation damage has the potential to revolutionize cancer treatment and various other fields. This innovative approach could lead to significant advancements in medical therapies and even space travel. Future outlook and predictions. 1. Enhanced cancer treatment protocols. The integration of Dsup into radiation therapy protocols for cancer treatment is poised to transform patient experiences. By significantly reducing the side effects associated with radiation exposure such as tissue damage and inflammation, Dsup could enhance the quality of life for cancer patients. We may see clinical trials within the next 5 years, leading to widespread adoption of this treatment in oncology by 2030. 2. Broader applications in chemotherapy. Beyond radiation therapy, the protective properties of Dsup could extend to chemotherapy treatments. As researchers explore the potential of using this protein to mitigate DNA damage caused by various chemotherapeutic agents, we could witness the development of combination therapies that improve overall treatment efficacy and patient tolerability. By 2035, we might see integrated treatment regimens that incorporate Dsup as a standard adjunct in oncology. 3. Space exploration and astronaut safety. The implications of Dsup are not limited to Earth. As space agencies plan longer missions beyond low Earth orbit, the need for effective radiation shielding becomes critical. The use of Dsup to protect astronauts from cosmic radiation may become a reality in the next decade. By 2035, we could see astronauts equipped with Dsup-enhanced treatments to mitigate radiation exposure during missions to Mars and beyond. 4. Development of modified Dsup proteins. One of the challenges identified in the current research is the potential immune response to the native Dsup protein. Future studies will likely focus on engineering modified versions of Dsup that are less likely to provoke an immune reaction. By 2030, we could have several optimized variants of Dsup that are safe for human use, broadening the scope of its applications in medicine. 5. Expansion into other medical fields. The applications of Dsup might extend beyond cancer and space travel. For instance, there is potential for its use in protecting healthy tissues during other medical procedures that involve radiation, such as certain diagnostic imaging techniques. Additionally, its role in protecting against oxidative stress could lead to breakthroughs in treating neurodegenerative diseases and aging-related conditions. By 2040, we could see Dsup incorporated into therapies across various medical disciplines. 6. Regulatory and ethical considerations. As with any new medical technology, the introduction of Dsup will necessitate careful consideration of regulatory and ethical implications. The speed at which these treatments can be brought to market will depend on rigorous testing and validation processes. By 2035, we can expect a more streamlined regulatory framework for mRNA-based therapies, allowing for faster development and deployment of innovative treatments like Dsup. 7. Global health impact. If successfully integrated into standard cancer treatment regimens and other medical therapies, Dsup could have a substantial impact on global health. By improving patient outcomes and reducing the burden of treatment side effects, we could see a rise in survival rates and quality of life for cancer patients worldwide. By 2040, access to Dsup-based therapies might be a standard aspect of cancer care in both developed and developing countries. 8. Public perception and acceptance. The introduction of a novel biological treatment derived from a unique organism may initially raise questions about safety and efficacy among the public. However, as clinical evidence mounts and successful case studies emerge, public perception is likely to shift positively. By 2030, we can anticipate a growing acceptance of advanced biotherapies, including those derived from extremophiles like tardigrades. Conclusion. The discovery of Tsup's protective capabilities offers a glimpse into a future where cancer treatments are more effective and less damaging to healthy tissues. The potential applications of this tardigrade-derived protein extend beyond oncology, promising advancements in various medical fields and even space exploration. As research progresses, the next decade will be critical in realizing the full spectrum of Tsup's benefits, paving the way for a new era in medicine and human resilience against environmental challenges.